Alright, so this video is going to cover two objectives in Chapter 12. We're going to take a look at Objective 6 and Objective 7. And they kind of go together because before we can understand an action potential, we do have to understand a neuron at rest. And so we actually covered this in Part 1 as well. But remember that our membrane potential is just that charge difference across the membrane. And so at rest, when we have a neuron that's not really doing anything, there's no electrical impulses going through it at the moment. It's unstimulated. Remember that resting neuron has a membrane potential of about negative 70 millivolts. And what that means is that the inside of the membrane is more negative, like you see here. And the outside of the membrane is more positive, like we see there. Okay, so that's just going to be our voltage difference, and that measures about negative 70 millivolts. So once again, the inside is negative and the outside is positive. And so what we call this is the membrane is polarized. Okay, it has poles, positive and negative poles. I also want to remind you about your ions, and that the sodium is much higher outside, as you can see, and the potassium is much higher on the inside. And so how do we maintain this resting potential? It just happens. This is just what it is. And so that resting membrane potential of negative 70 millivolts is maintained by a combination of these sodium-potassium pumps that are actively pumping out 3 sodiums and pumping in 2 potassiums, as well as those leak ion channels. Now remember that molecules will naturally move from high to low through these leak ion channels. There are more potassium than there are sodium. And so just to kind of show you why is it more positive out here and more negative in here. Well, first off, you have the sodium-potassium pump. And so you have 3 positive ions coming out, those sodiums, and you have only 2 positive ions going in, those potassiums. So that's the reason why sodium is higher on the outside and potassium is higher on the inside. But you also see you have more positives going out. Now, in terms of the leak ion channels, sodium is higher on the outside, so your sodium is going in to the cell. Your potassium are higher on the outside, and so, on the inside, and so your potassium are going out. But there's a lot more potassium leak channels. So once again, you have more positives coming out than you have going in. So as a whole, we're maintaining that positive outer membrane and negative inner membrane, because there are a lot more positive molecules outside than there are inside. And so this resting potential just is, and these proteins keep it around that negative 70 millivolts. And so Objective 7 is all about generating those action potentials. And so what's going to happen is we're going to have to change those charges across the membrane. And so what's going to happen is signals are going to come into the neuron. So remember, the signals are going to be coming from your sensory information. They're going to be coming in to the neuron from a sensory receptor or from another neuron. And those signals are going to be entering the dendrites of our neuron, our receivers. Now, that signal is going to, if it's strong enough, it's going to be converted into an action potential. And this is going to occur at that axon hillock in what we call the trigger zone. Now remember, not all stimuli will trigger an action potential. It does have to meet threshold. And so at rest, our starting point, we have the outside is positive and high sodium, and we have the inside is negative and high potassium. These leak channels are open, the sodium-potassium pump is active, we are sitting at negative 70 millivolts, and the membrane is described as polarized. So this is where we're starting. Now, once an action potential is going to begin, okay, the stimulus is going to arrive. It does have to meet threshold. That's about that negative 55 millivolts. If it's not strong enough, there will be no action potential. And so how do we actually generate an action potential? So notice that your stimulus comes in, that voltage is changed. What actually causes the action potential to happen? And it's going to be something called depolarization. Depolarization. Essentially, we're going to flip the charge on the membrane. So initially, it was positive on the outside and negative on the inside. Now, when this stimulus comes in, and it changes the voltage of the membrane, it's going to affect these voltage-gated sodium channels. And so sodium is going to rush into the cell, and so these positive ions are going to rush to the other side into the cell, and it's going to flip that charge. So when the sodium rushes in, it's going to cause the outside to become negative and the inside to become positive. Sorry, I messed up there. So depolarization is going to occur when that charge is flipped, and that charge is flipped when sodium rushes into the cell through those voltage-gated ion channels. And so the inside becomes more positive, the outside becomes more negative, and now we're looking at a voltage of about a positive 30. And so over here on the graph, you can see that that voltage is changing. This is what we call depolarization, and that's happening because sodium ions are rushing into the cell. And so here we had at rest, sodium's on the outside, potassium's on the inside, that negative 70. When that stimulus came in, it triggered these voltage-gated ion channels. Sodium is going to rush into the cell, and now we are going to have positive on the inside, negative on the outside. Now that doesn't have to stay like that for very long, because as these voltages are changing, they're affecting those voltage-gated ion channels. And so shortly after that depolarization occurs, repolarization is going to happen. And once again, we have these voltage-gated ion channels that are going to be involved. So those sodium channels are going to close because that charge is moving along the axon. And so as it moves along, those voltage-gated channels are going to close. So the voltage-gated sodium channels close, but that change in voltage does trigger the voltage-gated potassium channels to open. And so potassium is going to move out, okay? We also have the work of the sodium-potassium pump. And so between all of this, we are going to see that membrane potential restored, and it's going to become more negative again on the inside. So essentially, repolarization is going to try to take us back to that negative 70. Now, it actually goes a little bit further than the negative 70. It's called hyperpolarization. So those sodium channels stay open as those, the potassium channels stay open as the sodium channels are resetting. And so we actually get a little bit more potassium moving than what we need to. And so that's what causes this little dip here. We actually go a little bit below resting potential before we get completely restored. So that takes us to about negative 85 millivolts, but that's just for a very short period of time. And then we return to rest with the help of the sodium-potassium pumps. Those voltage-gated potassium channels are going to close, and we're going to go back to the work of the sodium-potassium pump and those leak ion channels, and that's going to maintain that resting potential of negative 70. So here we had, over here in our graph, we had our resting potential. The stimulus comes in, it meets threshold. Depolarization happens when sodium rushes in. Repolarization happens when potassium rushes out. We go a little bit under rest with hyperpolarization, and then it levels back off again. So here's another picture. This one actually kind of shows you what's happening with the gates. So the sodium channels open, that's what causes depolarization. Sodium channels close, potassium open, that's what causes repolarization. Here we have that hyperpolarization, and then the membrane potential is going to go back to rest with the help of sodium-potassium pumps. Now over here we can see how it propagates down the axon. So when this area is depolarizing, very quickly after, okay, that depolarization is going to be moving this direction, and shortly after that movement of the depolarization, we're going to have the repolarization occur, and that's just going to keep happening all the way down that axon. And so just kind of an overall summary, we started at resting potential, which is maintained by the sodium-potassium pump. We refer to the membrane as polarized during that time. The stimulus is going to occur, and that's going to trigger those voltage-gated sodium channels to open, which is going to lead to depolarization. And you can see that in depolarization, sodium is going in to the cell. Now, shortly after that, those sodium channels are going to close, potassium channels open, potassium start moving out, and that's going to lead to repolarization. We go a little bit lower than resting potential because those potassium channels stay open a little bit longer than they need to. And then with the help of the sodium-potassium pump, we go back to resting potential. And all of this is happening in a fraction of seconds. We don't even have to think about it. It's automatic. These are moving so quickly along that axon. Now the action potentials are all or nothing. If that threshold is met, that action potential is going to be produced, and it's going to be constant all the way down the axon. So the number of action potentials and the strength of those action potentials is going to be steady all the way down the axon. However, if the threshold is not met, then there will be no action potential at all. So it is all or nothing. Now there is a slight refractory period. So once that action potential is produced, it's no longer really sensitive to a stimulus in that very short period of time, and that's what we call this the refractory period. And so this is what keeps those action potentials from going both forward and backward. They're always going to be in a one-way direction. It's because they're not open to new stimuli as it's undergoing that action potential in that moment. So we have what's called the absolute refractory period, which is where there's complete insensitivity. So even if another stimulus were introduced, it's not going to generate another action potential. Now once we kind of get into that hyperpolarization zone back into getting close to rest, we do have what's called the relative refractory period, where if there was a stimulus that was strong enough, it might initiate another action potential, but normally we have this period of time where no other action potential can be generated. It's too busy trying to take care of what's going on in this one action potential to be prepared for another one. Now these action potentials are moved along the axon. So remember, they're generated here in the axon hillock, and then they're moving this direction along the axon. That's what propagation is. Now propagation can be affected by whether or not that axon is myelinated. The more it's myelinated, the faster it's going to be. It can be affected by the diameter, how wide that axon is. The bigger it is, the faster that action potential will travel. And in general, when things are warmer, they move faster. So the action potential will be quicker in warmer temperatures over cooler temperatures. So when it comes to the myelination, if you have a myelinated axon, that propagation is going to occur by what we call saltatory conduction. And so literally we have a jumping. Okay, so remember that the myelin sheath, it's going to insulate, but it's also going to speed up that propagation, because instead of moving all the way down the axon, it's going to jump from one node of Ranvier to the next. And so that allows it to move a little quicker like we see here. Now if you have an unmyelinated axon, it's going to move a little bit slower because it's having to move its way all the way down the axon. It's still fast, but it's going to be slower than what it is in saltatory conduction. When it comes to diameter, your larger axons are going to equal faster action potentials. Typically your larger diameter axons are also myelinated, which is going to speed them up even more. We'll see this in our motor and sensory neurons that have quick responses, like in our reflexes. Now your type B and type C are going to be smaller in diameter, and you're going to see less myelination as you go. And so the smaller and less myelinated, the slower those action potentials are going to be. And you're going to see these involved in your autonomic nervous system, in those automatic kind of responses that we don't have to think about, things like digestion and our blood vessel changes. We don't really have to think about those. They don't have to necessarily be quite as quick in their response. And so here we just have a quick comparison of graded potentials versus action potentials. So remember that a graded potential, these were just your different types of changes to the membrane depending on the strength of the stimulus. You have weaker stimuli that would lead to a lower graded potential. You would have a stronger stimuli. These graded potentials are what are going to possibly lead to your action potential if they're strong enough. So you could have some that are excitatory or some that are inhibitory. Now in order to reach the action potential status, it does have to reach thresholds, but a graded potential doesn't have to meet a minimum. they can occur whether they're weak or strong. Whereas an action potential is only going to happen if it reaches threshold, okay? The stimulus and the graded potentials can vary, whereas your action potentials, it has to be strong enough to meet threshold, and if it does, it's all. If it doesn't, it's none. The graded potentials fade out, but the action potentials are going to be constant along that axon. You don't really have a refractory period, but you do have one with action potentials. Okay, so all cells can experience these graded potentials, but only neurons and muscle cells can experience the action potentials. All right, and that's going to finish this video, and we will do our last one over Objective 8 next.